Okay, so thank you for joining for this session of this NSF Wednesday Colloquium. I am Jyotishman Dasgupta, the NSF Colloquium Coordinator, and I welcome all of you for those who have joined through YouTube as well as through this Zoom transmission. Okay, thank you for joining for this session of this NSF Wednesday Colloquium. I am Jyotishman Dasgupta. Okay, and I would like all of you to sort of recognize that this particular session of the Wednesday Colloquium is very special because it deals with this year's Nobel Prize in Sciences, the Nobel Prize in Physiology and Medicine, Chemistry and Physics, all of which will be summarized by our resident experts at TIFR. As we all know, this Wednesday Colloquium series is very traditional, as old as when Bhava started it, the start of this institution. And so it holds a lot of rich culture in discussing very important scientific topics that bother all scientists across all of us, across different disciplines. So for the Physiology and Medicine Prize, Professor Maitreyi Narasimha has kindly agreed to summarize finding the hepatitis C virus part of it. For chemistry, Professor ASR Koti is going to talk about the molecular scissors, how sharp are they? And for physics, we have two eminent speakers who are going to talk about both the theoretical and the experimental side of the Nobel Prize for this year. And the talk is titled Revealing the Dark Side. And that is given by Professor Gautam Mondal and Professor Saurabh Chatterjee. So as usual, we have reserved 30 minutes for each session for each of the prizes. For the chemistry and medicine, we have 25 minutes of talk time and 5 minutes of questions. For the physics one, we have, since there are two speakers, we have distributed the time as 15 minutes plus 15 minutes of talks and 5 minutes of questions. So with that, I would like to first welcome Professor Maitreyi Narasimha to talk about finding the hepatitis C virus. So Maitreyi, I'm going to unshare my screen and you can share yours.
Okay, thank you, Jyotishman. I am going to talk today about this year's Nobel Prize in Physiology and Medicine, which recognized the discovery of the hepatitis C virus, the second causative agent of blood-borne hepatitis. Since we're all sort of thinking hard about the coronavirus pandemic that's caused a lot of disruption to our lives, I thought I would use this opportunity to provide a little bit of a historical background into how one actually links an infectious agent to a disease. But before I do that, I would like to say that the work that the Nobel Prize this year recognized was the work of two Americans and an Englishman, Harvey Alter and Charles Rice, from the National Institutes of Health and the Rockefeller University, respectively, and Michael Houghton from the University of Alberta in Canada. And I would also just like to present this list that I compiled from the Nobel Prize website that documents the Nobel Prizes that have been awarded for the discovery of infectious agents or therapeutic interventions against diseases caused by infectious agents since the inception of the Nobel Prize. And there have been 111 prizes awarded in physiology or medicine, of which 14 of these listed here have been specifically awarded or the identification of a disease causing germ or microorganism, or therapeutic interventions that have allowed us to combat those diseases. And as you will see, the very first Nobel Prize in Physiology and Medicine was awarded to von Behring, who who sort of developed an antitoxin against diphtheria, which children today still get vaccinated against. And the subsequent years, prizes were awarded for the discovery of agents causing uh, malaria, or mechanisms that by which malaria is transmitted, and also for investigations and discoveries in relation to tuberculosis and trypanosomiasis, which is sleeping sickness. So you'll see that the very first Nobel Prizes were in fact awarded to the identification of causative infectious agents, uh, disease causing infectious agents. The inclusion of viruses in this list, viruses as causative agents of disease, came somewhat later, I would say from the 1950s, where yellow fever virus was the first virus discovered to cause disease in humans, and subsequently the uh, the human immunodeficiency virus, human papilloma virus, and now the hepatitis C virus were also recognized by Nobel Prizes. And in between, you will see there were uh, drug interventions that were also awarded with a Nobel Prize. So finding the agent that's responsible for an infectious disease has been an important theme in the award of the Nobel Prize. And many of these discoveries of finding the causative agents have the flavor of a very good whodunit mystery story. And I think I'd like to share some of the work that has led up to linking specific microorganisms to disease, obviously with a focus on the hepatitis C virus, but beginning with a general introduction on how you link germs to disease. Um, so if you look at a view that prevailed uh, during the Middle Ages, it was thought that there was a miasma theory of disease, which attributed disease, including diseases like cholera, malaria, and plague, to the inhalation of bad air that was thought to sort of live in or surround sick people. And so it was not uncommon to see doctors wear outfits like this that might look somewhat familiar in the present time, to ward off bad smells which they thought were responsible um, for disease. They believed that diseases weren't sort of transmitted from person to person, but were transmitted somehow through the air that came off from around a sick person. It really took Agostino Bassi from Lodi in Italy to propose that contagious diseases, could be caused by germs, and he was one of the first people to propose what we now come to call the germ theory of disease. And he developed this actually first by studying a peculiar disease that he noted in silkworms. And so the focus of his life's work was to keep silkworms and the silk industry healthy. And he was interested in asking questions such as which caterpillars, which are really these silkworm larvae, succumb to disease. Uh, how are diseased caterpillars related? How does disease in these caterpillars progress? And can one predict which caterpillars would get sick so that silk farmers could intervene before one sick caterpillar became an epizootic? Again, I think these are things that many of you are now sort of it's all in your awareness because of the coronavirus pandemic. And his research actually led him to discover that this particular disease, which is called muscardine of silkworms, could be reliably transmitted by simply transferring the fungal spores from a dead silkworm onto a healthy caterpillar. And so he showed for the first time that you could actually transfer disease from a sick organism, in this case a silkworm, to a healthy organism by simply moving the fungal spores from one to the other. And obviously, this had a huge impact on silkworm breeding, but more importantly, it had a huge impact on our understanding of disease, and it led to interventions that that are again all too familiar to us today. The first of these is the use of disinfectants to clean contaminated insects and equipment. You could call this sanitizing. The separation of caterpillars to prevent disease transmission, you could call this physical distancing. The removal of infected caterpillars before they became contagious, you could call this isolation or quarantine. But importantly, the proposal that humans must also be similarly susceptible to diseases caused by germs. So, in a way, I would say already back in the 1700s, we were aware of the fact that you could get disease by simply transferring microorganisms from a healthy animal to a sick animal. And we had interventions in place to try and curtail the spread of diseases caused thereby. Um, but it was really Louis Pasteur who sort of cemented the idea of what we as students learn as the germ theory of disease. And he showed uh, that microorganisms can mediate the afflictions of not just silk. He also studied another disease in silkworms called pebrine, but he also showed that the spoiling of milk and wine and beer, and also diseases that affect humans, can be caused by microorganisms. And he sort of laid to rest the idea that fermentation, which was one of the things he was interested in, happened by spontaneous generation of a series of chemical reactions, um, by showing that fermentation actually is carried out by a living organism. In addition, and somewhat unrelated to this talk, he also created the first vaccines for a number of other diseases that are caused by microorganisms, and these include cholera in the fowl and anthrax, a major disease of livestock that today has taken on a different dimension because it's been used in germ warfare, but importantly also against rabies. Um, and I want to now move on. I'll skip the bit about Edward Jenner here, but I want to move on to Robert Koch, who really laid the foundations of how we link a microorganism to disease. And he was actually, I'd say, a disease detective extraordinaire, because he identified the causative agents, not just of cholera and not just of tuberculosis, for which he was awarded the Nobel Prize in 1902, but he also identified the causative agents of cholera and anthrax. And some of his work on cholera happened actually very close to home in the Grant Medical College in Bombay, but also in Kolkata. So, in principle, he was worthy of receiving 3 Nobel Prizes, but he got one for his discovery of Mycobacterium tuberculosis as the causative agent of tuberculosis. Um, but what perhaps was conceptually more important was uh, his establishing a framework to link germs and disease. So he stated 4 postulates that he said must be satisfied in order for us to be able to say that a particular microorganism, be it a bacteria or a virus or a protozoan or any other kind of parasite is the cause of a particular disease. The first of these states that the microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. So, if these are the organisms, you should see them in a diseased animal, but not in a healthy animal. You should be able to grow or isolate these organisms from a diseased animal and grow them in pure cultures. And again, you should not be able to do that from a healthy animal. And if you take the pure cultures that you grew outside the animal and inoculated a healthy animal with this culture, that animal should now become sick and have the same features of illness as the original animal from which you isolated it. And finally, that if you should be able to re-isolate this organism from this second sick animal, um, and show that it's identical to the organism that you'd identified in the first case. Um, so these four postulates are still rigorously followed when one is trying to establish a link between a causative agent and a disease. And I'd just like to point out that Koch also attempted to make a vaccine against tuberculosis but failed. Um, and this became a bit of a scandal and subsequently prompted rigorous rules for testing vaccines. But we still use something like tuberculin as a test of exposure to tuberculosis. So, having said this much about the link between how you establish a link between a microorganism and a disease, I will now move on to hepatitis, which essentially means liver inflammation and is derived from a combination of Greek words for liver and inflammation. As I will tell you later, it's mainly caused by viral infections, although we know that alcoholism, toxins, and autoimmune diseases can also cause it. And way back in the 1940s, it became clear that you can get two kinds of hepatitis that are the result of infection by microorganisms. Um, the first type, hepatitis A, many of us may have had when we were somewhat younger. It's transmitted by polluted water or food and has generally little long-term impact on a patient, although you can be sick for 3 weeks, which is pretty bad. The second, which is the focus of today's talk, is transmitted through blood and bodily fluids and represents a much more serious threat because, in addition to a sort of short-lived acute condition, by acute I mean soon after infection, it can also lead to a chronic condition. By that I mean a long-lasting condition that can lead to the development of liver cirrhosis, which is also something you see in alcoholics, but importantly also liver cancer. Um, and the reason this particular type of hepatitis is particularly problematic is because you can harbor the virus in your body and be seemingly healthy for a very long time, and then show up serious complications like cirrhosis or cancer later. And it's been shown that bloodborne hepatitis is associated with a lot of morbidity and mortality and causes more than 1 million deaths worldwide. And so it's a health concern on the scale comparable to AIDS and tuberculosis. And the way you can get blood-borne diseases of any kind is by sharing injection needles, for example, by drug addicts, or by the reuse of inadequate sterilization of medical equipment in hospitals, or accidental needle sticks in healthcare workers, transfusion of unscreened blood and blood products, and sexual practices that lead to exposure to blood. So, hepatitis C virus is not the only virus that causes bloodborne hepatitis. In fact, the first virus that was shown to be associated with uh, hepatitis was in fact hepatitis B, and this was an accidental discovery that was made by Baruch Blumberg through his discovery of what is called the Australia antigen. Baruch Blumberg was neither a virologist nor an epidemiologist, but he'd like to call himself a medical anthropologist who was interested in understanding why members of different geographic and ethnic groups varied in their responses and susceptibility to disease. And so he came up with this clever idea that blood transfusions, people who have been repeatedly exposed to blood transfusions, would have in their blood protein variants that were different from their own. And this would incite from their immune systems an antibody response against to these new variants. And so to test this hypothesis, he reacted blood samples from such multiply transfused individuals against the blood from normal individuals from whom he had collected all these blood samples. And in so doing, he unexpectedly discovered an infectious agent for hepatitis B by finding that an antigen found in Australian Aborigines was also found in a New York hemophiliac. And he used a simple method which students of biology will know as octolony double diffusion, which relies on antigen antibody reactions identified by the formation of uh, by by immunoprecipitation, whose nature can allow you to determine whether you have identity, partial identity, or non-identity. Use this simple sort of method to try to identify um, to to do these reactions. And I just want to say that um, it was interesting that Bloomberg worked in a unit that was called Geographic Medicine and Genetics. And he continued his research on the genetic basis of differences in susceptibility with Harvey Alter, who is one of the laureates of this year's prize, which Harvey later termed his most important research collaboration. Um, and I won't describe how Harvey described it, but I describe the work with Bloomberg. But when Barry Bloomberg moved to the Cancer Research Institute in Philadelphia, he asked Harvey Alter to join him, but Harvey apparently wanted to get more clinical training in hematology. And people have said, I have 5 minutes, yeah, people have said that he would have shared the Nobel Prize if he'd accepted Barry's invitation. So, what Harvey Alter did do was to continue along the lines that he'd started with Barry Bloomberg. And in so doing, he found that he observed that there was hepatitis in patients who did not test positive for an antigen derived from the hepatitis B virus, right? And this he found this in a worryingly large number of cases, and this led him to conclude that there must be another agent besides hepatitis B that must be responsible for this disease. And he knew, because of tests with the hepatitis virus, A virus antigen, that it could not be hepatitis A either. And so he termed this illness non-A, non-B hepatitis. And using blood from these hepatitis patients, Alter was able to show that the blood was sufficient, human blood from hepatitis patients could transmit the disease to chimpanzees. And then subsequent studies also demonstrated that this unknown infectious agent that could transmit disease to chimpanzees had the characteristics of a virus. Um, and so then the question became, okay, if it's another virus, what kind of virus is it? And this is where Michael Houghton came in. He was then working at a pharmaceutical firm, Chiron, and he decided to isolate the genetic sequence of the virus. And to do this, he actually uh, took DNA from infected chimpanzees, or he created a collection of DNA fragments from chimpanzees that had been infected with human hepatitis patients. He argued that some of these fragments would come from the chimpanzee genome, but some would be from the unknown virus. And he made these DNA libraries in an expression vector so that they would produce proteins. And then he screened this library using antibodies that came from patients who had non-A, non-B hepatitis. And following a comprehensive search, he identified one positive clone and then went on to show that this clone was derived from a novel RNA virus belonging to the flavivirus family that he then called the hepatitis C virus. And this allowed the presence of antibodies in chronic hepatitis patients, then strongly implicated this virus as the missing agent. Um, So, in his own words, he said, they were forever trying to improve sensitivity and trying different approaches to try and identify hepatitis, what the cause of this disease was. They tried 30 or 40 different approaches over 7 years, screened hundreds of recombinant clones, and after 7 years, they found one, right? So the question then became, okay, there was this newly discovered virus, but could this cause hepatitis all by itself? And this is where Charles Rice came in, and along with other groups working with RNA viruses, he identified a part of the viral genome that is important for its replication, and showed that injecting this cloned virus into the liver of chimpanzees actually produced changes, pathological changes resembling what you see in humans with hepatitis. And this was therefore the final proof that the hepatitis C virus alone could cause the unexplained cases of transfusion mediated hepatitis. So therefore, we now know that in addition to the hepatitis B virus, the hepatitis C virus is an important causative agent of blood-borne hepatitis uh, that um, can can range in severity from a mild illness lasting a few weeks to a serious lifelong illness. We know that it can affect 70. It's actually quite a global health concern, um, and because of the discovery of the virus that causes it and our knowledge of what the virus encodes, we now have antiviral medicines that can cure more than 95% of people with hepatitis C infection. However, there's still no effective vaccine against hepatitis C, and research in this area is ongoing. I'll skip the bit about hepatitis C virus cell biology, um, but just say, since I'm running out of time, why this is a big deal. The Nobel Committee called this a landmark achievement in the ongoing battle against viral diseases, and said that because of their discovery, it was possible to devise very sensitive blood tests for the virus that have virtually eliminated hepatitis that results from transfusion of blood and blood products in many parts of the world. It also allowed the rapid development of antiviral drugs that, as I said, have been able to cure more than 95% of the patients. So, in a way, you can think of finding the hepatitis C virus as a chain of discovery that happened over a long period of time, and that included the isolation of and identification of the virus, understanding the mechanisms by which it can be transmitted, and also mechanisms of its infectivity. And I'd like to end with a few statements from the laureates post winning the Nobel Prize that all have a sort of common message. Michael Houghton said that he thought scientists had become a bit blasé over the last several decades, that we know how to stop viral infections because we've stopped polio, or we've curbed TB with vaccines. And he said that we became a bit over casual, that we could deal with COVID in this particular case easily. And I think obviously we've learned we cannot, right? And he also goes on to say that what we need for COVID is a vaccine. and that in order for us to get there, the emphasis of most governments around the world should be that we need more investment in virology, in infectious disease and immunology at the basic research level, but also to invest in translating it fast. Harvey Alter had this to say about the discovery of the hepatitis virus. Uh, he basically said it's a good story for a kind of non-directed research where we have a hypothesis, but you have no idea of where you're going to go, just looking to see what caused post-transfusion hepatitis. And he also said that it could not have been done anywhere else because it took so long to come up with something you didn't really expect to find. And the message is that it's important that you don't always know that important message is that you don't always know where you're going. Nowadays, research is directed and so has to come up with the drug fast. But at the NIH, they allowed me to just go my way. And, and Charles Rice has said that the activity around the world on SARS-CoV-2 virus is breathtaking and it's changing the way science is done. It shows you what can be done if people really mobilize and work together and bring different expertises to a common problem. So I think I will end there. I probably overshot my time and can take questions later.
Thank you, Maitri. Thank you very much. I would like to say that we have 2 to 3 minutes for questions, so we can take immediate questions from the Zoom audience here. Questions? Okay. So I just wanted to ask one thing is how so I mean, what are the basic similarities or dissimilarities between the A, B, and C types in the molecular sense?
Okay, they're actually quite dissimilar. They all belong to different families of viruses. So they're actually not at all related. Um, there's also a D and E now. So
Right.
there's 5 hepatitis viruses, and the D and E sometimes are seen as co-infections with let's say some of the other hepatitis viruses, but really they're not related at all. They're
Okay.
really different viruses.
Okay. I see Shudipto has raised his hand. Shudipto, can you unmute yourself and ask the question?
Yeah, fantastic talk, Maitreyi.
Thank
I
you.
have a question. So we are dealing with SARS-CoV-2, and very early on, it was sort of promised by everybody that in a year, maybe a year and a half, a vaccine would be available. If I look at hepatitis C, a vaccine is not yet available. Yet, if
Yes.
I look at the total number of people dying from hepatitis C, even in 2020, it's likely to be larger than people who would be dying by SARS-CoV-2, by COVID. Now,
Yes.
is it something that we know in scientifically about SARS-CoV-2 that makes this statement possible that we'll have a virus in, I'm sorry, a vaccine in a year, which is not true yet for hepatitis C?
Actually, I don't think myself qualified to answer that question, but I would say I think SARS-CoV-2 has somehow uh, raised hopes and expectations of people that sometimes seem to me a little um, over ambitious. We still don't have vaccines for many viral diseases that have been problematic. I don't know. I'm not sure. I think the last I read there were 300 and some 40 uh, candidate vaccines for SARS-CoV-2 and some fraction of them in phase 3 trials. And I don't know if there's any specific reason other than the sort of unprecedented pace at which research has progressed. Maybe again, because this is an economical problem more, well, it's also a medical problem, but because of how much it's affected our economy, it's also provoked much stronger response from the community than I think some of the other things did. So I would say maybe the pace at which research can progress. Now, I don't know if there's anything specific about the virus
Okay.
prompts us to see that.
Okay. A quick question by Vivek. Vivek, maybe a quick question.
Yeah, it's very quick. So you said that this virus stays in the healthy body for several years and then suddenly it becomes active and the person becomes ill. So
Yeah.
what could be the reason for such a inactive period and then suddenly it becomes active?
I'm not entirely sure what activates it. I will need to go back and check. But this is common to both hepatitis B and hepatitis C, despite the fact that they're very different viruses, and the cell biological mechanisms by which they cause hepatitis are also quite different.
Okay.
Okay.
Thank you.
Thank you. We will have a round of discussion after all the sessions end. So maybe we can take this again. Okay. Thank you, Maitreyi. Thank you for this very nice talk and for all the difficulties that you're in. Thank you for doing it and agreeing to it. Thank you. Okay, could you just unshare? Yeah, Koti, are you ready to share? Yeah, perfect. So we have the chemistry prize being given by Professor A.S.R. Koti. Koti? Koti, you are muted.
JD?
Yes, now we can hear you.
Yeah. And yeah, you see my slides, right?
So, yeah, you can start, Koti.
Hi. Hi, good afternoon, everyone. And I'm going to talk about so this year's chemistry Nobel work. I have to summarize the entire work in 3 words. It's basically molecular genetic scissors, you know. So basically they have developed a nice chemistry biology based tool which can be used, okay, which acts as a scissors at molecular level, and it's very sharp. So basically it can cut at a specified position on a DNA strand or RNA strand. You know, the pictures I'm showing is basically exemplify that. And this year's chemistry laureates are 2 women. Okay, this is the first time 2 women are sharing a chemistry Nobel. That's really great. And the first laureate is Emmanuel Charpentier from France. And another laureate is Jennifer Doudna, American, okay, from Berkeley. And why they are awarded the Nobel in chemistry? The Nobel citation says that for the development of a method for genome editing. So basically this is a discovery plus further development so that you can actually use this in for uses, okay, at molecular level in both living and non-living systems. Emmanuelle Charpentier, she was born in 1968, France, a French woman. She got PhD from Institute of Pasteur in 1995. She did most of the work which was recognized by Nobel is done at Umeå University in Sweden. She is currently a director of Max Planck Institute of Science of Pathogens in Berlin, Germany. So by training, she's a microbiologist. She has always been interested in pathogenic bacteria, why they are so aggressive, why they develop antibiotic resistance. Can we really find ways to treat, okay, and overcome this resistance so that we can actually do something about their infections? Second laureate, Jennifer Doudna. She was born in 1964, American, PhD from Harvard Medical School in 1989. And then she's a professor at University of California, Berkeley for a long, long time. And she is also HMI investigator. Her expertise is in different kinds of RNA. She is known for RNA interference. She's a biochemist. She is interested in proteins, RNA, protein-RNA interactions. So the work for which their work has been recognized is basically taking existing tools from nature, from living systems, and then modifying them so that you have a tool which can be used at will, okay, to apply to any DNA strands or RNA strands. To sum up, that's basically programmable genetic scissors, a tool for rewriting the code of life, okay, in living systems. Mind you, this discovery is very, 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 very young, just 8 years old. The key papers came in 2011, 2012, I'm going to talk about. And then the research has been really explosive in last 8 years. And people who work with basically DNA, RNA, or DNA, RNA dependent proteins, more or less they touch at some level this what's called CRISPR-Cas9 gene editing technique. Okay, so I'll also going to explain what this acronym means. So the picture shows that basically this CRISPR is RNA shown in red, and then blue is basically a double-stranded DNA, could be coming from any living thing like bacteria or, for example, from us. And then what is shown in orange is a protein called Cas9. Okay, it's basically two component system, beautifully two molecules, just Cas9 enzyme and CRISPR RNA will give you specifically cleave a DNA at a specified position. Okay, so background. So since the first structure of DNA proposed in 1953 by Watson and Crick, we know that how actually two DNA strands actually pair with each other. What are the specific bondings? You know, there are bases called ATGC, and then G only pairs with C and A only pairs with T. Just basically that information coming from experimental data of Rosalind Franklin, beautiful X-ray crystallography data on double-stranded DNA, and model proposed by Watson and Crick, gives rise to basically more or less how this structure can be really think of as a blueprint, how this structure actually can transfer information from DNA, which is inherited by offspring from parents, to a second level molecule called RNA, which is a cousin of DNA, and which can be further translated into proteins. So if you have to think a bacterial cell, for example, like E. coli, which is in your gut, okay, has 1 million base pairs approximately. We have, on the other hand, 1 billion base pairs, okay, 3 billion base pairs. And all the information which is encoded in this genome, okay, in our billion base pairs or million base pairs in E. coli, translate finally into proteins, and which finally carry the work, everything you can really think of, digesting food, walking, talking, seeing, listening to this talk, hearing to this talk, etc., are done by proteins. And we know that roughly eukaryotes have about 20 to 22,000 genes, and we don't have much information about all these 22,000 proteins, for example. So we want to really know. So how do you know that? Then how do you understand? So selectively, either you knock out a protein, or you knock in a protein, or you modify a protein by a mutation, okay, and then see how the organism actually behaves. Okay, this can be done in bacteria and mouse. But finally, you have to change it. You have to make the change in DNA only. Okay, so there are tools which have been developed since 1970s, come under what is called genetic engineering, have been used to regularly modify bacteria, plants, mouse, etc. For example, like bacteria can be modified to produce a drug molecule in high quantities. Plants can be modified so that they can withstand harsh weathers, and they can live against parasites. Are using mouse systems so that we can understand the disease progression of a disease, etc. But however, the methods are available, but they are so cumbersome, sometimes you can't really use for mouse. Even if you use, the result takes really long time, few months to few years. Even after the end of few years, you might not even know the change you wanted to make in DNA did not really occur. But however, with the development of CRISPR-Cas9 by Charpentier and Doudna, now we can actually accelerate this process. And for example, a mutant mouse can be created in weeks to just few months. Okay, so that's basically why this discovery or the development is very, very important. So what is CRISPR-Cas9? Okay, so let's go back about 30 years. Okay, in 1987, there is a Japanese group, okay, working with bacteria, E. coli, or gut bacteria, and then trying to amplify its genome, okay, trying to copy. they copied certain sections, and accidentally they copied a section where actually they found repetitive sequences, means the same sequence which I'm showing here, double-stranded DNA, occurs multiple times, but separated by what are called spacer sequences. These sequences are different from each other. There is no comparison, but the repetitive sequences are identical. And then few years later, another group from Netherlands again found a similar pattern in another pathogenic bacterium, okay, Mycobacterium tuberculosis. Again, repetitive sequences separated by spacers. Spacers are different from each other, but repetitive sequences are identical. In the same year, again, another Spanish group found in Archaea, okay, which are kind of cousins of bacteria. Even in this Archaea, they also found similar patterns. Okay, so this is a kind of mystery at the time, that why there are repetitive sequences are occurring, multiple repeats, and separated by spaces. Okay, so then later, after finding this information from different organisms, people put this into the acronym. These repetitive sequences are clustered together. Like I said, bacterium has 1 million base pairs. Okay, so but these are clustered in small section in that large genome, and these are regularly spaced by spacers, so regularly interspaced, and these small sections of this repetitive DNA are actually palindromic, means that one section can complement with the other part of the same repeat. So comes together, gives rise to this acronym called cluster regularly interspaced palindromic short regions called CRISPR. That's basically the origin. But then later studies showed that this DNA, double-stranded DNA, having repetitive sequences and spacer called CRISPR, can be transcribed into RNA, okay, like I showed you in the central dogma, but not further. So that RNA doesn't really result into protein. So it's not a gene, true gene. And also another discovery made was that not only this DNA is transcribed into single RNA strand, but which further matured into shorter fragments consisting of one repeat and one spacer. Very interesting observation. Okay, so this is roughly around 2002. And further discoveries showed that there are certain other elements in the close proximity of CRISPR called Cas genes. Cas means CRISPR associated genes. So these genes means that they transcribe finally and translated into proteins, okay, Cas proteins. But they are always found very near to CRISPR, okay, in that large genome of bacteria. And no one really knows why. But people know that because it's translated into protein sequence. If you know the sequence, you can actually do homology modeling. And then people discovered that these proteins are actually nucleases, means that they can bind double-stranded DNA, they can unwind two strands, and then cleave these strands. Okay, so that's the property of nuclease. So that gives rise to CRISPR-Cas. So basically, bacterium has this CRISPR-Cas. The question is that how does actually builds this CRISPR-Cas in the system? Okay, so whenever a bacterium is invaded by a virus called phage or bacteriophage, basically viruses infecting bacterium. Okay, so leaves a DNA inside this for invasion, and then bacteria are very clever. Okay, so they're actually copying whenever a new virus enters them. They copy a small stretch and put it into their memory, integrate into their own genome, so that this genome is actually copied, passed on to its basically daughter cells. So that's why the spacer sequences are different, because they're coming from different viruses. Not all viruses are identical, right? So again, gives rise to that. That's how actually bacteria developed a kind of immunity system. This is kind of ancient immune system you can really think of. And then somehow these Cas genes have a role in this. Okay, so this bacteria has basically this memory of this CRISPR-Cas coming mainly because of viral invasion, and they would want to keep in memory so that next time the same virus attacks them, they want to cleave this DNA. So this CRISPR-Cas has somehow again detects if the same virus enters. Okay, the question is how. Okay, so there are many hypotheses at the time put forward. So the CRISPR has a repetitive sequence. There are Cas genes. These Cas genes gives rise to proteins. Proteins somehow cut a small segment of invading DNA or RNA from the virus, put it into their memory. Okay, so then this DNA is transcribed into RNA. RNA gets matured. We don't know why, because people have observed both single-stranded RNA and short pieces of RNA. And then somehow this finally gives rise to what is interference. Whenever the same virus again attacks the bacteria, so these pieces of RNA somehow detect that and then cleave, acting as molecular scissors, okay, acting on DNA. How this CRISPR RNA is matures into these small fragments is not really known. That's where actually the research of Charpentier comes. The first key path-breaking publication in 2011 in Nature, where actually she discovers what is called tracker RNA. Okay, so there are a lot of different types of RNA are there inside the cells. Okay, interfering RNA, small RNA, ribosomal RNA, tRNA, and whatnot. But she found that there are certain RNA elements which are large in amounts. Typically, that's not natural. And somehow she figured that the sequences of these RNA, short RNAs, very similar to CRISPR DNA. Okay, shows that there is some link. They have to be real linked. How, we don't really know. But her discovery showed that how this short stretches of RNA called tracker RNA, where do they exist on the genome of bacterium? And she showed that this sequence is very close to the location or the loci of CRISPR-Cas genes. Okay. And again, she showed the directionality, which stand or not. These are only details, but it's a beautiful paper where actually explains that, discovers a new type of molecule called tracker RNA. And then she shows with many experiments that how this tracker RNA actually somehow linked to these repetitive sequences, which I showed. Okay, the sequence is partly identical, matches with this. So her experiments, done many different experiments, shows that, so once the CRISPR RNA is translated into DNA into RNA, this RNA, now this tracker RNA short stretches actually match, and then another enzyme or the Cas genes produce Cas9 protein, come and bind every tracker RNA complemented with the CRISPR RNA, bind, and then another nuclease comes and cleaves, and then you have genetic scissors. So this is a genetic scissors. So where the scissor is basically the Cas9 protein, which can cleave DNA. Okay. And during that time, that itself actually explains how the single-stranded CRISPR RNA actually matures into a shorter RNAs. So in 2011, after she published this paper, she meets Doudna in a conference in Puerto Rico. And then over a coffee table, they discussing with each other there. So Charpentier is a microbiologist, Doudna is biochemist and RNA expert. So Doudna was also actually parallelly looking at CRISPR-Cas systems in a different organisms. Charpentier did all the work inside a bacterium called Streptococcus pyogenes. It's basically pathogenic bacteria. And she is interested in basically how the further mapping of this matured RNA gives rise to basically the cleaving ability or the scissors ability of this system when a new DNA inserted by, for example, let's say a virus. Okay, so now most of the elements are known. Basically, you need a Cas9 protein, you need a tracker RNA, you need CRISPR RNA, and you need basically viral, for example, DNA. So if you know the systems well inside a cell, then you should be able to replicate this in test tube. Very simple principle in biochemistry and biophysics. So Charpentier and Doudna collaborate, and then they do their first experiment. They put in a test tube a Cas9, matured cRNA, and a viral DNA corresponding to matured cRNA. And lo and behold, nothing really happens. The experiment is total failure. Okay, viral RNA is not cleaved at all. But then after long thinking, many discussions, they do a small modification. They repeat the experiment, but they add now a small amount of tracker RNA, which was not there earlier. Lo and behold, now everything really works perfect. A small change shows that tracker RNA is extremely important in doing this CRISPR-Cas9 work. Without tracker RNA, you can't really do. So this is basically published in a second key publication just 1 year after 2011. Basically, their Nobel Prize is based on just 2 publications, okay, 2011 Nature and 2012 Science. Here, interestingly, they do everything in test tube experiments, many different controls, small stretches of DNA, taking either from a virus or plasmids, which are available in molecular biology labs, or you can really think of everything which can be done in test tube. And they really understand how the Cas9 protein works. Like I said, Cas9 is a scissor protein. So scissor protein means that this is responsible for acting as a scissor, and it acts on a DNA strand, for example, in this case. Okay, and they also know that this Cas9 has 2 nucleus domains, basically 2 domains which can independently cleave DNA. So if you submit a double-stranded DNA which matches with CRISPR RNA, okay, that's very important, then this Cas9 can actually cleave double-stranded DNA and they can actually separate. Okay, so now they have done a lot of experiments where actually they can mutate one domain and then only one strand is cut. They can mutate another domain, only other strand is cut. They even know which domain is responsible for which strand, because in DNA both strands are different from each other, right? Because only one strand actually complements with the CRISPR RNA. Okay, so this is beautiful paper again, nicely demonstrates the tool in vitro. Okay, yeah, in vitro. And then they actually even simplify, okay, a lot of other complications which are there in bacterial system, they simplify. They even made simple CRISPR RNA and tracker RNA into single molecule called now guide RNA, as a small guide RNA, and then Cas9. So this is basically a 2-component system, okay? So which can be used in test tubes, which can be used in live cells or live organisms. Okay, so this is latest. What is the development? So CRISPR RNA, CRISPR-Cas only cleaves DNA, nothing else. But then you can actually use bacterial, let's say, system further, where you can actually repair this double-stranded break using a template so that you can actually insert a gene of your interest or create mutation. Or you can modify your Cas9 scissor protein so that you can insert single site mutation, or you can add some other complexity that you can actually even design the entire DNA sequence so that you can actually add a new sequence without coming any template. Okay, so these are explosive research in all these this. So I'll give you 3 examples how basically the CRISPR made into different research areas. The first example in plants. So we know that plant breeding, so typically takes a lot of time, you know. So if you want to get a good breed, you have to really wait for years of research, and then very small changes really occur in genes, and then selectively you take one gene and go on further. But using CRISPR and Cas9, you can do that actually very fast. You can really see that 2014 to 2018. So CRISPR-Cas has been applied to almost all fruits you can really think of: apple, oranges, gourd, okay, grapes, you name it, okay. And there are many different ways of actually using this. So you can take CRISPR-Cas, because there are different types of CRISPR-Cas in bacterial systems. You can actually use them to different types of plants: mustard plant, rice, soybean, tobacco, you can really think of. For the purpose of, for example, to provide resistance against, let's say, any diseases, mold, okay. For example, let's say drought areas where less water is there, so you can actually create genes where they are beneficial for the plant, and so on and so forth. So explosive research in plants. Second example is that application of CRISPR-Cas9 in cancer therapy. So this is a very recent example, okay, published in February, showing that from the patients, for example, like blood cancer and connective tissue cancer, which are not really responding, these are refractory cancer, they are not responding to treatment. They have taken the T cells from these patients, modified certain elements, using CRISPR-Cas, very simple Cas9 guide RNA, change specific elements, and then put these modified cells back into patients. So these T cells actually survived few months, up to almost 9 months, and then provide some assistance, okay, to fight basically the cancer. So that's basically, so CRISPR is already into modifying cancer therapies which are beneficial. But there are a lot of controversies because such a beautiful technology. So biotech industry will jump on it right away. Okay, it's $1 billion industry already. It will be even few more $1 billion industry in future. Okay, so there's a big battle between University of California, Berkeley and Broad Institute fighting for the patent. You know, Broad Institute, the professor and the postdoc replicated CRISPR-Cas9 in eukaryotic cells and then filed a patent. Initially, they were actually awarded the patent, but then Berkeley filed a case, and then there's a fight still going on. Okay, so the fight started, for example, 2017. Round 1, basically Broad wins. Round 2, no, I think both are really fighting. I think this is the latest, 2 months ago, it is September 11, 2020, the latest round in the CRISPR patent battle has an apparent victor. Says that, okay, Broad can use the prior usage of CRISPR-Cas9, but University of Berkeley also will get some benefits, okay? So, but the fight is going on, okay, ongoing battle. What is future? Okay, so the future is actually bright in terms of using gene editing, let's say, especially curing inherited diseases like sickle cell anemia or Huntington disease, etc., where actually you know that these diseases are because of the mutations which are inherited. So if we know that these mutations exist at the embryonic level, in principle, you can actually change this mutation back into wild type or normal or healthy mutation, so that the embryo develops into a healthy baby without inheriting the disease. That's really bright future. Gene-edited organisms, they're already there. For example, bacteria, mouse, it's not a problem. But gene editing babies, okay, that's a big ethics question, right? And obviously, people have been really thinking. So this is a statement by Jennifer Doudna, says that the power to control our species' genetic future is awesome and terrifying. Deciding how to handle it may be the biggest challenge we have ever faced. Okay, so obviously we know that it's very easy to use this technology even in test tube or in live cells to change the DNA at will. But then just after this comment was in 2017, and then just about a year after that, you really have a gene-edited babies. Okay, so this scientist from China, Hong Kong, Jiankui, without getting any approval, he took a human embryo and he played with a certain gene which is supposed to be causing HIV, or which is supposed to help enter HIV virus. He actually played with the gene in the embryo and then implanted in a woman, and then finally they have a twin babies. You know, these are like gene-edited babies. This is really without really thinking about what is the consequence. Okay, so we have to really think very carefully. Lulu and Nana, okay, gene-edited babies. But leaving the talk on a positive note, obviously CRISPR one can use for beneficial. So this is the example. So Indian perspective, we have been thinking on how to actually use our resources to fight this pandemic, especially COVID-19 or SARS-CoV-2. So we want to really use any tool at our disposal if it can really help. Obviously, CRISPR, which is actually derived from bacteria fighting viruses, obviously you should be able to use somehow for fighting, okay, SARS-CoV-2 or COVID-19. So Shavik's lab from IGIB, CSR lab at Delhi, has developed a CRISPR-based tools called Feluda test. So if you have a RNA extracted, for example, let's say a patient, okay, so from the nozzle swab, you just extract RNA. If you can submit that RNA to this CRISPR-based test, which is very fast, in about half an hour to 1 hour, you should be able to see whether someone has this virus or not. It's a paper-based test, shows that, for example, if you see 2 bars, it's positive, 1, no one. So just about 2 weeks ago, okay, CSR Director General Shekhar Mande said that this tool should be available soon. Okay, so this is in collaboration with our commercialized wheat artisans. Okay, with that, let's congratulate Emmanuel Charpentier and Doudna for their exciting discovery and development, which has been useful. So, though it is very young, it has been useful in last years, and it's really transforming both biology and chemistry equally. Thank you.
Thank you, Koti. Something really beautiful work got represented in the by the Nobel Committee. And thank you for doing a fantastic job, Koti. It was very clear. Thank you. We have some questions already in the chat box, Koti. I'll just take one quick question. There is a question by Alak. I will take Alak's question later. But first, there is a student question, Sujit. How the specific recognition of DNA by the tracker RNA occurs here? Is it similar to complementary base pair recognition or anything else?
Yeah, that's exactly complementary base pairing. Okay, so basically, sorry if I have to go back to a few slides. Yeah. Yes, definitely. So what you see is the tracker RNA, which has exactly same sequence as DNA. Okay, in part. Okay, some parts don't match. Okay, this is where actually the palindromic hairpin comes into picture. So otherwise, this black part is actually identical. It's purely based on complementarity.
Okay, and since there is a little bit of a time, I'll take Alak's question. Has CRISPR technology been applied to research such as salinity-resistant strains of rice?
Yeah, so yes, I think it's difficult to cover. Basically, like I said, the research has been really explosive. I think people have been really using, so from the rice perspective, I think pest resistance or salt resistance, how exactly rice can survive, for example, let's say in heavy metal containing water conditions, etc. It has been there.
Okay. Okay.
People have been really doing.
Okay. Okay. Thank you, Koti. I think we will move on to the physics prize, and we will come back another round at the end for a discussion.
Thank you.
Yeah, thank you. So now I invite Professor Gautam Mandal to kindly share the screen and keep his video on as well. Gautam, are you on the video? Yes. Okay. Now you are muted. You are muted, Gautam.
Can you hear me now?
Yes.
Okay, very good. So thank you, Jyotishman. And so I got my 15 minutes shot at revealing the dark side. So here we go. Okay, so this is about the Nobel Prize in Physics 2020. So I'll talk about the theoretical part of Roger Penrose. And hopefully we will do something better than the gentleman in the cartoon who just sees something completely black. All right, so the Nobel Prize in Physics this year was divided. One half was awarded to Roger Penrose for the discovery that black hole formation is a robust prediction of the general theory of relativity. And the other half is going to be covered by Shorav, so I'll leave it to him. So in order to understand the implication of Penrose's work, we have to go back to history and a little bit of prehistory, in fact. So as early as the end of 18th century, John Michell from England and Pierre-Simon Laplace from France, they said that, well, if a star exists with the mass-radius relation, which is the following, that the radius is less than a certain critical radius, R0, is equal to 2gm by c square, where g is the Newton constant and m is the mass of the star and c is the speed of light, then not even light will escape from it. So in modern parlance, this is called a black hole. So these gentlemen were the first discoverers of black hole solutions, so to say. So, and in particular, probably the fatherhood should go to John Mitchell. All right, so how does the math work out? So it's something very simple. So and I'm not going to do too much math here, but this is something that I can probably do. So let's say that consider the Earth, okay? And let's try to find out the minimum velocity with which a projectile must be thrown so that it barely manages to escape to infinity, okay? In that case, the total energy at infinity will be 0. Its potential energy will be 0 because it's inverse 1/r. And kinetic energy is 0 because it barely reaches there. So total energy is 0 there. So therefore, the total energy to begin with must be equal to 0. So you can plug in your formulas and you will get this fact. The relation between the radius from where it was thrown and the escape velocity. Okay, so that's the relation. And if you take the escape velocity equal to the speed of light, you will get the characteristic length that Mitchell and Laplace got. So for reference, the escape velocity on the surface of the Earth is 11 kilometers per second. And if you replace the Earth by the sun and shrink the radius from what it is to 3 kilometers, then you can easily check that the escape velocity does become the speed of light. So in other words, a solar mass object with a radius of 3 kilometers or less becomes a black hole. Now, both Mitchell and Laplace regarded this more as a theoretical curiosity than something that could possibly exist in nature. Why? Because such a star would be enormously dense, okay? So in particular, the density would be 10 to the power 19 kilogram per meter cube, whereas the densest material known at the time was gold, which had merely 10 to the power 4. Okay, so it was unimaginably dense, and it was not pursued any further. However, looking back in retrospect, that was not the main point about the extreme densities. The point is that for such strong gravitational forces, in retrospect, Newtonian gravity, which was used to do these calculations, was not the right framework anyway. So it had to be replaced by the general theory of relativity. So then the question becomes that are black holes allowed in general relativity or are they in fact predicted by general relativity? Okay, so that's the question. The answer came from a battlefield. So Einstein's equations of general relativity, so I just wrote something here. You don't have to really know what I have written here, but these are the equations, which are some complicated partial differential equations involving 10 metric coefficients, which are 10 functions of 4 variables, 4 arguments. And so these are hard to solve. In fact, even Einstein got to solve this only in an approximate manner in his 1915 paper. And however, within barely a month, okay, the answer, as I said, came from a battlefield given by Karl Schwarzschild, who was ignoring all the heavy gunfire that was going around him and was working on Einstein equations and found a solution, exact solution. Okay. So what was the solution? The solution was, so these are the metric components given in polar coordinates. Once again, I will tell you the physics of these expressions, but just note that there is a characteristic length scale, which is the same length scale as was found by Laplace and Mitchell, somewhat surprisingly. So it is 2gm by c squared. And so this is the gravitational field of a spherical star, exterior gravitational field outside of a spherical star. Now, if it so happens that the radius of the star is the same as this characteristic length scale, this characteristic length scale is called the Schwarzschild radius or the Schwarzschild horizon radius, etc. We will explain why it is called the horizon radius. Okay, so if the star has a radius which is less than the Schwarzschild radius, then something very weird happens. So let me tell you that one of the effects of one of the lessons from general relativity is that the clocks run at different speeds at different places. So in particular, if you were standing on the surface of the Earth, then the clock at the feet of your at the bottom of your feet will run a tiny bit slower than the clock which is at the top of your head. So in case this kind of a thing happens, that the star has a radius which is less than the Schwarzschild radius, then the clock sitting at the horizon here would run infinitely slowly, literally infinitely slowly. So the time will just freeze at that point. Okay, so if you start from far away, then the clock will start going progressively slower. The other way around there's another way of saying this, the same thing is that if you start from here and go outward, then the clock runs progressively faster. So it's infinitely slow here, then it's some finite, finite speed, etc. So if you consider two flashes which are sent out from the horizon, at an interval of 1 second, then at a local time of 1 second, then it reaches some finite radius such as here at an interval. So the relative the interval between the two flashes will be infinite seconds, literally. So there's a formula given here, and the basic point is that the GTT, which is the time component of the metric, it vanishes at the radial coordinate r equal to rs. So that is so therefore the light that was sent later, it never reaches. So basically light does not escape from the horizon at all. And this is what, so we got the GR form of the result that Laplace and Mitchell wanted to get. So light doesn't escape from the horizon. An alternative way of saying the same thing is that as a star collapses with its radius eventually shrinking below Rs, the Schwarzschild radius, the light from it becomes more and more redshifted because of the same thing. Okay, the clock slowing, more and more redshifted till it becomes so faint that it becomes an invisible hole in space. Okay, so that's a black hole. The name was given by Wheeler. So extreme space-time award. Okay, and it's predicted by an exact black hole solution of general relativity. Okay, so there we are done. Not quite. Einstein was very impressed with the exact solution of Schwarzschild, but doubted if this could represent a real object formed from the real collapse of an astrophysical star. Okay, does it happen that way? The answer this time came from a ship. Okay, where Subramaniam Chandrasekhar was taking a ship ride from India to England and working out the equations of stellar evolution. And he found that there was a mistake in the earlier calculation in which, the earlier calculation of the energy versus radius was something that allowed for a stable radius, okay, always, for a white dwarf. He found that in his calculation, if the mass of the white dwarf was taken to be greater than a certain limiting mass, which we call these days the Chandrasekhar limit, 1.4 times the solar mass, then there is no stable radius at all. Okay, wherever you place that the star, it will go towards the collapsing solution, which is the r equal to 0. Of course, in real physical thing, what happens is that as it progressively shrinks, it becomes a neutron star or a black hole and so on. But black hole becomes actually from the it comes from the realm of mathematical physics to real life here.
3 minutes.
Yeah. Hello. Oh, 3 minutes. Oh, Jesus Christ. Okay. So Chandrasekhar's life was, work was based on relativistic QM, and it was met with skepticism. So I'm not going to go into the details of that, but In 1939, some more evidence in favor of black holes was found by Oppenheimer and Schneider, who constructed an exact gravitational collapse solution from a spherical distribution of pressureless matter. Okay, so here you have an exact solution again, this one. And so what it showed was that there was a singularity. So it had a central singularity. And so once again, because of the spherical symmetry, people thought that it was too idealistic. And in real life, what will happen is that the slight departure from spherical symmetry will grow in time and prevent the collapse from occurring. Okay. Now, this discussion was analogous to the discussion of initial singularity of spherical cosmologies. So in fact, the Oppenheimer and Schneider solution, so this solution used a collapsing cosmology inside and the Schwarzschild radius outside. So if the collapsing cosmology is possible here, then the gravitational collapse into a singularity will also happen. So these two equations, these two questions became the same. Whether in the absence of a spherical symmetry, the challenge was that in the absence of a spherical symmetry, whether cosmological collapse can happen, which is like a rewind of Big Bang, and a gravitational collapse can happen, as in black holes. Now, the first hint of this, the fact that it can happen without any assumption of spherical symmetry, was it came from the first ever singularity theorem, which was proved by Amal Kumar Raychaudhuri at 1955, using his equation, Raychaudhuri equation, derived from Einstein equations, which basically says at what speed do neighboring trajectories of particles come together, okay, in the presence of gravitational force and matter field, which are such that they satisfy some energy condition, which are very reasonable energy conditions for ordinary matter. This object, okay, so the assumption was that this R mu nu, U mu, U nu, which are some components of the Ricci curvature tensor, they have to be positive. This is called a strong energy condition. And under that, what happens is that if you start with these, these orange geodesics, these are trajectories of material particles. And if they start contracting to begin with, then there's no way that they can bounce back as in here. So this scenario is not possible by Raychaudhuri equation. On the other hand, what does happen is that they converge into a single point, which is called a focus. Okay, so no spherical symmetry is assumed here, and no, so it's in fact a local statement. So these are infinitesimally separated geodesics. So here's the math for this, that the relation between theta dot and theta. So theta is the expansion factor, is the area of cross section, the rate of change of the area of cross section divided by the area. That's called a theta. From this equation, you can derive this result. Okay, that there is focusing. And if Raychaudhuri equation, so now if these are actual fluid particles, okay, then you are done, because when it's converging into a focus, there is clearly going to be infinite density, because the volume becomes zero here, and so you have a singularity. However, we want to talk about the spacetimes themselves, so spacetime geometries in which we want to talk about the behavior of geodesics, that is to say the behavior of straight lines in gravitational fields, but not necessarily the paths which are traversed by actual fluid particles. So these are possible paths, okay, which are geodesics which characterize the nature of a space-time geometry. Okay, and for black holes you have to use null geodesics, and for cosmologies you have to use time-like geodesics. So for these, so the focusing theorem does not imply singularity in these situations for the space-time geometries. So do the space-time geometries collapse with that in mind? So we actually need new concepts. So what the role of Roger Penrose was a mathematician from Cambridge. He was an algebraic geometer who was drawn to astrophysics by Dennis Sciama, the cosmologist. And he brought in, from his experience of mathematics, many absolutely essential concepts called geodesic, such as geodesic completeness, closed trapped surface, etc. And using those, one could arrive at yes answer to both cosmological collapse and black hole collapse. Okay, so Jyotishman, I have minus some minutes.
Yeah, you're -2, but that's okay. Go ahead, finish it.
I have just I think two more slides, 2 or 3 more slides. Okay, so so now let me discuss the cosmological collapse, which is slightly easier to explain than the black hole collapse, which I'll take later on. The concepts are very similar. So how does one, using these tools, how does one prove cosmological collapse? Okay, so now, I have to just say one little thing here, that in terms of mathematics, this guy happens, the focusing happens if theta is negative to begin with. That is to say, the area is shrinking. Okay. To begin with, if the theta is negative, then in the future there will be a focusing, and looking into the, okay, so here it is. So suppose we assume the following thing, that our space-time has an initial value surface. Okay, this is a very reasonable assumption, that you would like to give your initial data at some surface at time, sort of the notion of some time t equal to 0, and then that should on the basis of that you should be able to predict what happens in the future. Okay, now it can be shown. Okay, so for lack of time, I cannot go into the details of this, that all points, if any point in the space-time, must be connected to this surface, the initial value surface, which is called the Cauchy surface, by a maximal causal path. This is an extremal path, so, and no such causal path can have a focal point. Okay, so that's very easy to show. This is by using Cauchy-Schwarz inequality, which is also called Twins paradox in general theory of relativity. And by then, so if there is a Cauchy surface, then every point of the thing this must be connected by a maximal causal path, that is to say some geodesic, time-like geodesic, and no such path can have a focal point. However, if you assume that all over this Cauchy surface, theta is negative, okay, not the same value perhaps, but theta is negative everywhere, okay, then there must be a focal point on every time-like geodesic. So you have a contradiction here. Therefore, what that means is that the such points P beyond this beyond the appearance of the focal point, okay, will simply not be allowed. Okay, so that means that there is a singularity that has appeared in the space-time. So this is what now I should explain what is meant by saying that theta is negative all over the place. Okay, so it means that you look back in time from our current cosmological slice, where you have an expansion looking forward in future. So looking backward in time, so therefore you have a contraction. So therefore you must have, by doing this logic, looking backward in time, you must have a collapse, and that collapse is basically the Big Bang. Okay, so it looks like a collapse because we're looking back in time. Okay, so in case of so this was by looking at the properties of what are called time-like geodesy, the paths of material particles. In order to look at the the black hole collapse, you have to look at null geodesics, that is to the properties of light rays, light paths, and so basically if you look at the Schwarzschild solution once again, what you can show very easily, that light cone gets flipped once you cross the horizon, that both the outgoing rays and the ingoing rays, so outgoing rays and the ingoing rays, both become ingoing rays here. Okay, so in your future you have always you are going towards the singularity. So this phenomenon, okay, was generalized by Penrose, that both the outgoing as well as the ingoing light rays become both ingoing. So that's called the closed trap surface. And what he showed was that if there is a closed trap surface, then this thing that people were afraid of, that or maybe secretly hoping that if you have a spherical lack of spherical symmetry, then somehow various paths will miss perhaps each other, and eventually there will be bounce and there will be no collapse. Okay, that does not happen. Okay, even locally, if this this closed trap surface means that singularities will appear. So as a result, we get the singularity theorem of 1965 of Roger Penrose, which says, building on the work of Raychaudhuri and using mathematical tools discussed above, he showed that once a collapse has progressed up to some stage so that a closed trapped surface has formed, that is to say, both types of rays are ingoing, and provided the null energy condition is satisfied. So that's another condition on the stress energy tensor, very reasonable conditions, and nothing can prevent a singularity. And that's what fetched him a Nobel Prize this year. And here we go.
Okay. Okay, Gautam. Sorry to rush you, but thank you for doing this from the very start, and very simple classical ideas. Thank you for doing that. And I will actually not take questions now, Gautam. We will take it after Saurabh completes the presentation.
Sure.
The experimental. So Saurabh, could you share your slide? Maybe Gautam, you could unshare.
I will do that. I will do it.
Are you seeing my slide?
Yes.
Am I audible?
Yes, you're audible. Perfectly fine. Thank you, Saurabh. Yeah, you can start.
All right. All right. Good afternoon, everyone. I will take my turn to reveal the dark side. And so, of course, I don't mean revealing dark side like this. I actually want to share with you something that's way more exciting. Some trouble.
Yeah. Is okay.
All right. So I'll be talking about the achievements of our latest superstars in physics, Roger Penrose, Reinhard Genzel, and Andrea Ghez. As the Nobel press release states, and Gautam mentioned, Penrose received the prize for the discovery that black hole formation is a robust prediction of the general theory of relativity. Genzel and Ghez received the prize for the discovery of a supermassive compact object at the center of our galaxy. So Gautam has already talked about Penrose's contribution, so I will not touch on that. I will talk about the achievements and contributions of Genzel and Ghez, including the existence of a supermassive black hole at the center of the Milky Way beyond any doubt. And this one picture is the culmination of about 30 years of hard work and groundbreaking advancements in observing technologies. It shows the measured and modeled orbits of stars very near the center of the Milky Way. All of them are orbiting around an invisible mass center, which we know today to be a supermassive black hole. That is a black hole of mass above a few million times the mass of the sun. But let me tell you the story from the beginning and the key stakes towards success. Before I dive into the main course of today's talk, I want to point out that black holes, which has always been fascinating for the general public, is now a darling for the Nobel Committee as well. 3 years ago, the Nobel Prize was awarded for the detection of gravitational wave radiation from 2 marching black holes. So this is the brief outline of what I want to cover during the time I have left. First, I will give you an ultra brief summary of the backstory of observations. Then I will talk a little about the strategy and hurdles to detect a possible black hole at the center of the Milky Way. Then I will talk about the breakthrough advancements in observational technology that made this discovery possible. And then I will simply show you the results. Throughout the talk, I may be taking some liberties in the way I explain things so that it's widely accessible. And apologies to the. So the first observational hints for supermassive black holes came from the so-called quasi-stellar objects, in short, quasars. These are extremely bright radio sources found in all sky surveys, first discovered back in the late 1950s. For a few years, there were no optical counterpart found for these systems, and it was not really clear what these systems are. However, the game completely changed with the discovery of the quasar 3C273 in 1963. Using the radio localization by Hazard et al., Schmidt found an optical counterpart. From prominent spectral lines, they also found that this source cannot be a part of the Milky Way. Instead, they found that this is at a redshift of 0.158. That is, it is several billion light years away from us. So once you know the distance and also can measure the apparent brightness of a source, you can estimate the luminosity. And it was readily understood that this is an extreme system with luminosity over 1,000 times larger than that of our entire galaxy. It was also found that this source not only is extremely bright, it varies its intensity on timescales of hours. So these rapid fluctuations strongly indicated that the engine driving this source is not only extremely powerful, but also must be very small and not extended. By 1965, Schmidt found many other such extragalactic quasars with distances extended to redshift of 2. And by this time, supermassive black holes as the driving engine for quasars became the most favored model. Interestingly, this modeling is the subject of another Nobel Prize given to Riccardo Giacconi, who explained the cosmic X-ray sources as accreting stellar mass black holes. The extreme brightness of quasars were modeled simply as an extension to these models. So, according to this model, brightness is coming from accretion of gas onto the central black hole. The gravitational energy released is converted to heat and radiated away. However, there is a limit to the rate of accretion of matter. At some point, the pressure from the emitted photons can become too large and either halt accretion or can even blow away all the matter that could have been accreted. This is called the Eddington limit. And the expression looks somewhat like this. So the luminosity is related to the mass of the accretor by this equation. This is basically photon pressure and gravity ratio. So this is the Thomson scattering cross-section. This is the mass of the photon. This is the velocity of light in vacuum, and this is the mass of the accretor. Okay. You can see that the limiting luminosity is directly proportional to the mass of the accretor. So let's look at some of the numbers for our 3C273 QSO. It has a mind-boggling luminosity of 10 to the 40 watts, and that gives a mass estimate of 1 billion solar masses. So another step was taken by proposition by Lyndon Bell. He proposed that these supermassive black holes inhabit the centers of many, if not most, galaxies. And the quasar phase is simply a phase in the accretion. And even if you don't find a quasar in a galaxy, there may be a supermassive black hole that simply isn't accreting enough. Okay? And then the natural question was, is there a supermassive black hole in our galaxy? In fact, a compact radio source called Sagittarius A star was detected at the center of the Milky Way However, at that time, the nature of this remained unknown. So let's think about how would you actually detect a black hole at all, okay? So imagine that you have a black hole of mass m, and there is some radius. This is called innermost stable circular orbit. Anything that goes in falls onto the black hole. So the idea is to find the mass enclosed in some small volume. In other words, an average density. So if you have a black hole like this of mass m and the ISO, and you can calculate the average density like this, and it becomes 6 times 10 to the 23 solar mass per cubic parsec for a 4 million solar mass black hole. So that's a very high density, okay? And then how will you detect this density? If you can follow stars that are orbiting these black holes, and hopefully a close-by star goes through, you can detect the orbit of a close-by star, and then you can model the mass of the central object that you don't see, and that mass must be enclosed within the pericenter of that orbit, okay? But for the Milky Way, this is a very, very challenging proposition, and this is why. So to understand why it's challenging, let's take a look at the zoom in movie of the center of the Milky Way, released by ISO, using observations by the Hubble Space Telescope. You can see that the center of the Milky Way is extremely messy. It is a crowded region with lots of stars, gas, and dust that obscure light, okay? So somehow you need to observe stars in that area, which is 8 kiloparsec away from us, and resolve the orbit and find some average density in a small volume, okay? So that's the challenge. So to measure stellar orbits close to the center, one needs to overcome all of these challenges. Let me just tell you what these challenges are. First of all, you need a very long baseline observation to follow orbits, okay? That rules out space-based observations. And then obscuration from interstellar dust will stop you from observing in optical, for example. And Gez and Genzel both use near-infrared observations to mitigate this challenge. And then it's an extremely crowded region of stars, so you need very, very high-resolution images. And to do that, you need very large telescopes. But the problem with large telescopes, large ground-based telescopes, is that this telescope is sitting under the sea of under the atmosphere, and turbulence in the atmosphere will smear the light that's coming onto your telescope. And this problem is the larger the telescope, the higher this problem is. So what did they do? So first of all, Gez used the Keck Observatory. This is a 10-meter class telescope located in Hawaii. Genzel used the new technology telescope initially, and later the very large telescope. Both are ISO telescopes located in Chile. As I already said, both used near infrared for their observations. And back in 1998, they did something really brilliant. This is called speckle imaging. So the idea is, so all this atmospheric turbulence is mirroring things at time scales of a second, okay? So if you take really, really short exposures, very short, sub-second exposures, then of course, each exposure will be really bad. You cannot go deep, but they will not be smeared. And then you can take thousands of these exposures. So this is one such exposure. You can take thousands of these exposures, And then using the bright stars as your guide, you can stack the all these images on top of each other, like they did. And if you then zoom in to this tiny square, this is the image they receive, right? This is the central 1 arcsecond square around the Sagittarius A star. So this is really brilliant. So using this technology, both of them measured the proper motions of stars that are nearby the Sagittarius A star, and then plotted that as a function of the projected distance from Sagittarius A star. And they found that these velocities show Keplerian drop-off as the distance goes, becomes higher. Okay, and at that time they measured that the mass is a little over 2 million solar masses, which later have been updated to 4 million solar masses. After 2003, another technique called adaptive optics became available to both of these groups, and that completely changed the game again. So the idea is basically, if you have a guide star, the light is straight line here, and that is going through the atmosphere, and it becomes distorted. But if you can also distort your mirror on the telescope at time scales that are relevant, that are smaller than the turbulence time scale, then you can recreate the undistorted image in your telescope. Okay? Just to give you an example how powerful this is, so this is observation of the same two stars. Only the left one does not have adaptive optics, the right one does. So what did we gain from between speckle image and adaptive optics? So adaptive optics allow you to give very long exposure. So that means you can do very deep observation. Okay, so that means you can detect fainter stars. In addition to that, you can also do spectroscopy. That allows you to measure the radial velocity. Remember before, they were observing the proper motion. So that is motion in the sky plane. So that gives you two directions of the velocity, two components of the velocity. And now the radial velocity is perpendicular to the sky plane. So now you can get all the three components of the velocity. Okay. So they did that for since 2003, and they have now measured the full orbits of one close by star to the Sagittarius A star and enough portion of other orbits of the other nearby stars. And this is basically the solution. And the orbit of S2 or SO2, that's this yellow one that has the full orbit resolved, that gives you the full orbital solution. So the orbital period is 16 years. Eccentricity is about 0.9. It is inclined to the sky plane at 46 degrees. The pericenter distance is 125 astronomical units. This means that the Sagittarius A star is about 4 million solar masses, and this is enclosed within at most 125 AU. That gives you an average density of 5 times 10 to the 15 solar mass per cubic per second. This is at least 10 times the density of any known extended bound mass distribution. So that settles it, that it's a compact object, it's enclosed in a very, very small volume. And that's why they got the Nobel Prize. Thank you.
Fantastic, Saurabh. Very, very dense ending. So, so I would now like to thank all the speakers for a very nice session of sort of discussion and presentation of the major highlights of what the Nobel Committee thought were fascinating and worth the prize. So I would now open the session to more questions. First, I will open the physics session. So I would request both Gautamda and Saurabh to be on the video. So questions for Gautamda.
Should I stop sharing?
You can, yeah, you can keep it at this point. It's a beautiful picture. So yeah. Questions? Acha, Amol has a question. Amol, you can come into video and ask the question if you can. Okay. If you,
Yeah, I just came.
yes.
So hi,
Yes,
Saurabh. The question was just what is the typical response time of adaptive optics mirrors that you
I
mentioned?
think it has reached microseconds now.
Okay.
Okay.
So it has, I think, many thousand actuators, and at least I don't know about the VLT, but cake is a honeycomb design. So it has thousands of these small honeycomb pieces that create the whole mirror, and each of these can rotate and do jiggling.
So that's much faster than atmospheric fluctuations.
Yeah.
Okay. Gopu has a question.
I think Arnab asked something.
Yeah, first you go. I'll take Arnab as well.
Yeah, I mean, so this is just to connect the first two parts. I mean, what Gautam talked about and what Shorav talked about. In the next 10 years or so, there will be at least 3 distinct billion-dollar observatories trying to ensure that this is actually a black hole as defined by Penrose by testing black hole no-hair theorem. But that's a saga in itself. So, more things are going to happen.
More things are going to come. Okay. Yeah.
Yeah, absolutely. I wish I could touch that, but it was a short time.
Yeah.
So just to mention the facility, so TNT, the 30-meter telescope that India is a partner of, is going to increase the resolution of this area significantly, and it will again give us a lot to learn. So actually, in addition to knowing about the supermassive black hole, there are open questions about the mass distribution of other stars and other different post-Newtonian effects and so on. That will be revealed by TNT. There is LISA, that the primary source of LISA is supermassive black hole mergers. So of course, that will be phenomenal. There is the pulsar timing array that is also going to detect supermassive black hole mergers.
Okay. Cool. I will now let Arnab ask the question to Gautam. I think Arnab, are you there?
Yeah, I just wanted to, Gautam said you'd say, why is it called the horizon? And I guess you were pressed for time, so you never came back to it. But just curious, why is it the horizon?
Yeah, I can come back to that without a slide, but then if necessary, I can go back to the slides. So basically, so there was this particular radial distance called the Schwarzschild radius, and there were two things that I showed in the beginning. I showed that because of the extreme time dilation, light simply doesn't manage to reach a distant observer, and because light does not reach from there, so that is why we call the horizon. We can't see anything beyond. We can't even see the horizon itself because the light gets infinitely redshifted, so it just loses all its energy. So we just see the thing black. But the main reason why it's called a horizon, in fact called the event horizon, is that nothing from beyond that place reaches us. No light reaches us. We don't see the stuff beyond.
Okay. Okay. There are two questions from students. One from Pradeep. Pradeep, can you unmute and ask the question?
Yes. So my first question is, which formed first, our galaxy or the supermassive black hole that is the center of our galaxy?
What came first? That is the question.
Should I try to answer?
Who would like to answer? Yeah.
I can.
Yes, sure. Yes, sure.
So that's an excellent question, and we do not have a definitive answer. actually the question as posed is slightly ill-defined, because the galaxies are not monolithic systems. Galaxies keep changing, they merge with other galaxies, and they keep forming. So, but if I can slightly change the question and say, did star formation in a galaxy happen first, or did a supermassive black hole form first? That we do not know the answer of.
Okay.
And my second question is, we assume that the gravity or the gravitational wave is constant everywhere in the universe. So what is the peculiarity that the gravity is so much strong in the singularity of the black hole at the point? Because is that the only gravity? Because if in the black hole singularity, everywhere gravity is, we know that we assume that's constant. So what is the reason that gravity is so much strong at the singular point of the black hole?
Who would like to answer that? Not very, yeah. Gautam?
Yeah, maybe I can give it a shot. So I'm not sure exactly what. So one definition of a singularity is that the the curvature tensor becomes, or some scalar formed out of a curvature tensor becomes infinitely large. But that's a technical question. So that answer. Let me tell you in terms of tidal forces. So basically, when you approach a singularity, let's say a human being with head towards the singularity is approaching the singularity, then tidal forces will simply tear his body off, because he would see a much stronger gravitational attraction towards the singularity than his feet. So this is one example of a tidal force. So he will be extended. His body will be extended more and more and more. And if he was, of course, infinitely flexible, then it will just keep getting infinitely extended. So that is one way of thinking about singularity. Of course, the way Penrose and Hawking, they discussed their singularity was a little different. That was about some kind of an end of space-time. Or at least certainly the end of some trajectories of possible trajectories of light. Suddenly they end. Okay, so it's not they don't commit to why it's ending. Is it ending because there's an infinite density? Is it ending because there's an infinite temperature? Is it ending because there's an infinite curvature? They don't say that. But certainly in the context of black hole singularity or cosmological singularity, you can just say that there we do know the situation is that it's infinite tidal force.
Okay. Well,
can I ask a question?
No, Pradeep, I think we can ask Sharanno, who is also wants to ask a question. Is that okay?
Yes.
Yeah, Sharanno, please can you?
Yeah, I had a question about this trapped minimal surfaces that Gautam sir mentioned. And before, when you were talking about Schwarzschild and all, there was a clean sort of way of at least pictorially representing that there is a horizon of some radius and so on and so forth. Right. Now, as far as I understood, Penrose elevated this to more generic situations where such a spherically symmetric situation may not be the case. Right. In that case, does a role of a horizon get played by this minimal surface business? And I mean, are these minimal surfaces unique in that sense that I can say that this is the largest minimal surface that I can make? Or
I mean the closed trap surface. Yeah.
Closed trap surface. Oh, sorry. Yeah.
Yeah. In a sense, it's the role of bending light, even the outward going, what would have been the outward going light is was being played by the horizon in case of the Schwarzschild black hole because of its perfect spherical symmetry. But the place where you have so this same phenomenon, if you just take this property of the horizon, then indeed the closed trapped surface is playing exactly that role. And what you can show is that if if it so happens that both beams of light, or rather I should say light paths, both the extremities of your light cone, they are bending inward, then you can, by applying these various other energy conditions and the kind of theorems that we did for the cosmology, you can show that locally, okay, for that particular in the infinitesimal neighborhood where you are talking about the light cone having this property, they will end up in a singularity. So you won't be able to extend those geodesics, those null geodesics beyond a certain point. Yeah, so it's a
Okay.
local.
Okay, Sharon, is that okay?
Yeah, I'm satisfied. Thank you.
Okay.
I mean, it's easier to draw it, but I guess there's no time. Yeah. So.
Okay. There is one question for the chemistry prize from Shujit. Restriction endonuclease can also cut the DNA in specific positions. So why CRISPR-Cas9 along with the tracker RNA becomes important in modern context to cut DNA? Is Koti there?
Yeah, I'm there. Yeah, it's a very good question. Yes, absolutely. Restriction endonuclease is actually also very selective to DNA sequences. But the problem is that you can't really have restriction endonucleases for all possible sequences. You have only a handful of restriction endonucleases, which we get from already existing living systems, for example, like bacteria. But what CRISPR-Cas9 offers is that using just one system, you have to just change the sequence of CRISPR RNA. Once you change this guide RNA in the guide RNA CRISPR sequence, then you can actually use, for example, let's say we have a billion base pair genome. Literally you can go anywhere you want. Our bacterial genome has million base pairs. You literally can go anywhere. But restriction nucleases actually offer very little small space, okay, which can be spanned across eukaryotic or prokaryotic genome. That's the limitation.
Okay. Are there any other questions or comments by any of attendees here?
Can I ask a
okay.
question?
Yes.
So my questions to Professor Koti sir, that if we say cut the DNA genome edit and slice it and we enter by CRISPR-Cas9 and another part of DNA incorporating in that DNA. So there may be possibility that the functionality of the DNA or the cell's ability to react with the disease, or let's say some sort of other cancer or disease can also be possible for genome cutting. Because if we did if we cut the DNA.
Okay, let me tell you. See, CRISPR-Cas9 only worries about cleaving DNA. It could be double-stranded, it could be single-stranded, it could be RNA. But it doesn't really say anything about after that, So after, for example, let's say if you cut DNA in living system, so the living systems will respond, they try to repair. There are majorly two different repair mechanisms inside, for example, in bacteria. So one of them will pop in. But other than that, it's like it doesn't really worry about anything else. It worries about only two things: selectivity, sensitivity. Its sensitivity is very high. It means that once it binds, it cleaves DNA. Specificity is actually really much better than, I would not really say 100%. There are something called off targets. Okay, but these are very few. Very, very few compared to any of other existing methods which have been, for example, in the market, let's say to edit genomes. So in that sense, CRISPR-Cas9 gives you much better, the best so far.
Okay. I think Shubha has also raised. Shubha, do you have a question?
I just wanted to just extend Koti's answer. Yeah. Is that also the answer to the restriction enzyme question? Because restriction enzymes, the very specific ones recognize 8 base pair bits of DNA, which after all are found, at some frequency in DNA, whereas guide RNA can be about 24 base pairs. So you have much more specificity because you can design whichever sequence you want. To find that same 24 base pairs multiple times is rarer than finding 8 base pairs.
Mm-hmm. That's very nice.
Yeah.
Thanks, Shubha.
Thanks, Shubha. Are there any other questions, comments? If there are none, I think Pradeep has ask one to Maitreyi. Is Maitreyi there? Can hepatitis B and C occur at the same time? Is
Can they occur at the same time? I guess in principle they can, and I think in practice, one of the things that's common to bloodborne diseases is that many bloodborne diseases, especially from transfusions, can coexist. So, for example, hepatitis B commonly found in AIDS patients. So I mean, I am sure they can occur at the same time. I don't see any reason why they can't.
Okay. Okay. And there is one final question. Dev Shankar had put it in the chat. I missed that. I'm sorry. What is the role of tracker RNA in gene splicing? Can you give a molecular insight? Koti, is that Koti? Yeah.
Gene splicing?
So this is what he has said.
Is he there?
Yeah.
Dev Shankar?
Dev Shankar? Or maybe he left.
Okay. Yeah. Well, as far as I know, this guide RNA doesn't really involve.
Dev Shankar, you are there? Okay.
Dev Shankar, you have to unmute.
I don't know if he's.
No, he's there. That green window popped up on his.
Okay, but he is not. I can't unmute him.
Koti, I think what he's trying to ask is what is the role of the tracker RNA in the final cutting of the DNA by the CRISPR-Cas9. So splicing is a different thing. He might be just trying to mention the cutting of the DNA.
Yes. Okay. So both Charpentier and Doudna have done a lot of control experiments. For example, if tracker RNA is not there, so then the binding of CRISPR RNA is very weak. Okay, it doesn't really bind strong. Second thing is that, so basically the domains, the Cas9 is a big protein, so the domains actually really wobble. They don't have a flexible structure. But unless tracker RNA is there, so which binds at a specific location and also pairs up with the CRISPR RNA, which defines shape. Okay, so without tracker RNA, even if you have complementary DNA RNA of CRISPR RNA, the Cas9 is not going to function. That's what actually Charpentier's second paper or 2012 science paper that will actually really show. It shows that it's the role played by tracker RNA is so important that without that, the machine scissors doesn't work. Though you have scissors protein, but it is not functional. So again, it shows that the role when they have done when they combined, tracker RNA along with CRISPR RNA, made a single element or two component system, saying that it's single guide RNA. So that CRISPR element, the tracker element is very, very, very important. So to place how the Cas9 binds, to help bind the viral RNA is absolutely important.
Yeah. Okay. Thank you, Koti. And thank you to the entire audience who have been sticking for this long till the end of this session. I really would like to thank all the speakers once again for doing this colloquium. This is the most awaited colloquium usually for the entirety of year. So thank you for the splendid talks, all four of you. And thank you for doing it through online medium, which is slightly different than what we actually typically get. And we would have taken all of you for a little bit of a tea or coffee and still let people mingle with you. I'm sorry for that, but I have passed on your email address to students as well as people in the Zoom window, and I will probably do that in the Zoom, in the YouTube as well. So, so thank you. Thank you, Gautam. Thank you, Saurabh. Thank you, Koti. And thank you, Maitreyi, for all the efforts.
Thank you.
And I will officially close this session. And just to tell you, we will return back next week with a sort of a talk in optics given by Yuri Kircher from Australian National University. I hope you can join us for that session on Wednesday, that is the 11th of November. Thank you. Thank you once again. And thank you.